Hello, everyone. Thank you all so much for joining our seminar today. We will begin promptly at noon Eastern Time. Hello, everyone. Thanks so much for joining. We will begin the seminar promptly at noon Eastern Time. All right, hello everyone. Thank you all so much for joining today. Welcome to the NOAA Omics Seminar Series. I am your host, Catherine Egan. I am the NOAA OAR Omics Coordinator, and I sit in NOAA's Office of Ocean Exploration and Research, or NOAA Ocean Exploration. I also sit on the NOAA Omics Working Group as a subcommittee co-lead. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We have established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside of NOAA. The seminar series takes place on the third Wednesday of every month, with some seminars scheduled outside of this time frame. I encourage you all to check out the website for more information. So the seminars from this series are all made available on YouTube and posted up on our website a few weeks after the initial presentation. I will drop a link to our website in the chat right now. Okay, and you can also reach out to me via our NOAA Omics email if you have any additional questions. So that's up here on the slide, noaa.omics@noaa.gov. As participants, you all are muted, but feel free to type questions into the chat box throughout the presentation. At the end of the presentation, I will read the questions to our presenter to answer. So with that, our presentation today is titled Understanding the Genetic Mechanisms that Facilitate Local Adaptation in the Genomics Era, presented by Dr. Wes Larson. So Wes, I am going to give you screen sharing permissions now. And while you are setting up your presentation, I am going to introduce you to our audience. Okay. So Wes is originally from San Diego, California, where his passion for the ocean and fish was ignited during countless hours spent on outdoor activities such as scuba diving, kayaking, fishing, and snorkeling, as well as deckhanding on a sport fishing boat. Wes received his BS in marine biology from the University of California, Santa Cruz, and subsequently his PhD from the University of Washington. During his PhD, Wes focused on applying genomic tools to study local adaptation of Pacific salmon and inform fisheries management. After finishing his PhD in 2015, Wes worked as the assistant unit leader of the USGS Wisconsin Cooperative Fishery Research Unit, where he led a research program that generated genetic information to inform fisheries management. In 2020, he started as the new program manager for the genomics program at the Alaska Fisheries Science Center in Juneau. His main research interests are applying genomic techniques to inform fisheries management and study local adaptation. So Wes, thank you so much for presenting today. I can see your presentation, and I will now pass it off to you.
Perfect. Thanks so much for that introduction, Catherine. And can you guys hear me okay?
Yep, we can hear you great.
All right. Well, thank you so much to everyone for being here this morning, or I guess close to afternoon, depending on where you're at. And I guess since that intro was so complete, I'll just dive right into it here. Let's see. All right. So first off, why should we care about adaptive genetic diversity? I want to start with a bit of an introduction, not getting right into the genomics, but taking a little bit of a step back and talking about why we should even care about preserving adaptive genetic diversity. So first off, I think it's a well-known kind of concept that in investments, the more stocks, bonds, houses, whatever you have, the less risk. You can even think about it like I'm a big skier, and so like with ski resorts, these big conglomerates have been buying up all the ski resorts, and then if there's, if somewhere has a bad winter, somewhere else has a good winter, it kind of evens things out. And so that's a pretty well-known concept in kind of finances, and it turns out that it's also very true kind of in ecological systems, and there's been quite a bit of work on this portfolio theory over the last decade or 15 years or so. So to kind of demonstrate this, I'm going to contrast two different systems where salmon live. Bristol Bay, Alaska, which is a pristine system that produces some of the largest, if not the largest, runs of sockeye salmon in the world, and the Sacramento River, which has been degraded over the last 150 years, and genetic diversity has been severely eroded. So, and we'll be getting back to this, but Bristol Bay happens to be the area where I did a lot of my PhD research, so it's definitely a near and dear to my heart kind of place. And so Bristol Bay is in the Bering Sea, kind of in Southwest Alaska. If you've seen Deadliest Catch, those boats fish sort of right around here on the Eastern Bering Sea shelf. So just to put it into some context. And Bristol Bay has 9 major rivers that produce salmon, each of them with many locally adapted and actually genetically distinguished or genetically differentiated populations. And this is one system, and all of these different dots are salmon producing rivers and habitats. And so over the last 5 years or so, Bristol Bay has had some of the largest runs of sockeye that they've ever had, even while a lot of other places are struggling kind of pretty mightily. And so this is a quote from Daniel Schindler, who's a big ecologist that works in the system. So one of the reasons Bristol Bay is so unique is that all of the habitat diversity is still here, all of that genetic diversity and life history diversity is still here. And it's interesting scientifically, but it's also important for the fishery because of all of this diversity stabilizes how many fish come back from year to year. So Bristol Bay is a system that has a huge amount of genetic diversity, a huge amount of life history diversity, and is producing some of the biggest runs of salmon in the world, even while so many different systems are struggling. And so to contrast that, we can move to the Sacramento River in California. And so the Sacramento River has been heavily impacted by humans since the gold rush in the 1840s, and continues to be impacted by things like droughts, urban areas, dams, and pretty much everything you can, hatcheries, pretty much everything you can think of that can imperil salmon populations. And so a really nice paper from Munch et al. Just came out a couple weeks ago in Global Change Biology, talking about kind of the loss of this portfolio of adaptive diversity in the Sacramento River and how that impacts the stability of fisheries and other things that depend on these runs. And so this is just a graph kind of showing where the historic habitat is. So over here, they're showing like where different runs of salmon traditionally spawned or historically spawned, and then now it's all truncated because of these dams over here. And so a little bit of context, there's these 4 different runs of salmon in the Sacramento River, but it's changed over time where, 150 years ago, a lot of these runs had pretty high abundances to now things are pretty much dominated by fall run, and most of those fall run are produced by hatcheries. And so here's a graph or kind of a diagram showing how this impacts fisheries. So essentially what's happened in the Sacramento River is that genetic homogenization in hatcheries has produces most of the fish from a single run, and that run has kind of synchronized life history characteristics. So they all outmigrate at the same time. They all kind of come back at the same time. And if those fish hit something in the environment that makes the a high mortality event, then a whole cohort can be wiped out. And so that's what it's demonstrating down here is like if there's a hot April or there's a situation where they hit the estuary at outmigration and there's not enough food, or it's hot when they're coming back, you can pretty much lose a whole cohort here. Whereas if you have an intact portfolio of life history diversity, even if one run has a bad year, there's all these different runs that are kind of spreading out the risk. And then even if these guys get have a high mortality event, you still have a stable fishery and a stable run due to all this different life history and genetic diversity. And so I think this diagram does a really nice job of kind of visually illustrating this concept here with spreading the risk out across life histories. And so contrasting Bristol Bay, the Sacramento River has had some really tough years over the last couple decades, including a couple years here from like 2008, 2009, and 2010, where there pretty much wasn't a fishery. And so the red here is this ocean harvest, and there was no fishery in 8, no fishery in 9, very little in 10, and a little bit more, but not much in 11. These 4 years, that's going to be enough to pretty much knock most commercial fishermen out of the game, to cause them to diversify or change the type of boat they use, etc. So that type of fisheries collapse for multiple years is pretty devastating to a commercial fisheries and commercial fishing industry. So hopefully I've kind of sort of shown you some reasons why adaptive genetic diversity is important. Again, to kind of review, so Bristol Bay has this really intact portfolio of adaptive genetic diversity, and even in the face of things like climate change or different environmental conditions, the runs are very stable because these different components kind of, some years it's a down year for one component, some years it's an up year, and through that they all kind of buffer the system. And then the run is still really good. Whereas the Sacramento River, obviously, they've kind of genetically homogenized. There's less adaptive genetic diversity in there. All the life history characteristics are kind of synchronized, and then you get these major boom and bust cycles, which can lead to huge fisheries closures. So hopefully you guys are with me here on that adaptive genetic diversity is important. And so kind of where my talk is largely going to focus is how can we identify adaptive genetic diversity and understand the mechanisms that create and maintain it. All right, so now we're going to dive right into the genetics and genomics, and I'll try and go here slow here because there's quite a few concepts that sort of build on each other. Okay, so first thing, before I start, I'm going to say FST a lot. FST is a measure of genetic differentiation. It ranges from 0 to 1, with 1 being very high genetic differentiation, and really anything over 0.1 is pretty high genetic differentiation. And I'm also going to show a lot of graphs with a lot of dots. Most of the time, the dots are genetic loci, which is a genetic marker in the genome. So in this graph, each one of these dots is a genetic locus. And the first concept here is that genetic differentiation is often heterogeneous across the genome. So there can be markers that are showing low differentiation, and there can be markers that are showing high differentiation. This is a graph from Limborg et al. 2012. This was on herring. And what they saw is that many or most genetic markers showed very low genetic differentiation, but there was a few that showed extremely high differentiation. And this is called an outlier test. So in this graph, the x-axis is support from a Bayesian model for a given locus being under selection. And over on this side of the graph is the area of the graph where there's the most evidence for loci being under selection. And so highly genetically differentiated loci are potential candidates for being under selection because they're kind of more diverged than you'd expect under neutral processes. Okay, so the next kind of thing to build on here is that genomic resources can help us understand the landscape of differentiation across the genome. So maybe 10, 15 years ago, 5 years ago even, people in the genetics field were doing a lot of these, what's called an outlier test. And so you get all your loci, let's say it's 100 or 1,000 or 10,000, and you use a statistical test to try and see if those loci are more genetically differentiated than you'd expect under neutral evolution. And so you figure out a way to draw a line here, and anything above that line you'd call potentially or putatively under selection. And so in this graph, the line is this. It's just sort of flipped in this graph. So anyway, these different colored markers would be candidates for selection. But the thing is, if we just do these outlier tests without any genomic resources, we don't know where these loci are in the genome. So they could all be found right next to each other, or they could all be found scattered across the genome. And there's kind of a big difference in that. If they're scattered, it's kind of hard to pinpoint exactly what's going on as far as like which genes are under selection, which areas of the genome are under selection. But it turns out that often these loci are kind of localized in what we call genomic islands of divergence. And I'm going to be using this term genomic islands of divergence a lot. Basically, it just means an area of the genome that's showing much higher genetic differentiation than the surrounding areas. This graph happens to be from Atlantic cod ecotypes. So there's migratory and stationary ecotypes of Atlantic cod, and most of the genome has very low differentiation for these ecotypes, but there's a couple spots on a couple chromosomes that show extremely high genetic differentiation. They happen to be inversions, which we'll get into later. But this was a paper with like maybe 1,000 loci genotyped with a chip, and they were already starting to see this pattern. Okay, so highly differentiated loci that are likely involved in selection are often found clustered together in islands of divergence. And so the next step here is to think about why. So I'm going to use kind of a simple diagram to kind of illustrate how these genetic islands of divergence may evolve. And so we're also going to be looking at a lot of these different of these plots where genetic differentiation, or FST, is on the y-axis and the genomic position is on the x-axis. That's kind of the same as this here. This will come up over and over again in the talk. And so here, these are just like kind of fake chromosomes, and I colored them kind of alternatively. Okay, so first off, if you go out there and you sample a single population of fish, and then you go out there, say you go and sample it 1 week, and then you go back and you sample it the next week, what you expect to see is little to no genetic differentiation. So across the genome, no differentiation. Okay, but what happens if the yellow population of fish starts kind of inhabiting a new area and starts kind of evolving a new phenotype? So we could call this early divergence, kind of early population divergence. Turns out what you see pretty often is that most of the genome still shows very low genetic differentiation, but there's a few areas, which we were calling islands of divergence, that show much, much higher differentiation. And these areas are likely involved in kind of adaptive differentiation. So they probably hold kind of the genes that are actually facilitating, phenotypic changes associated with selection and adaptive divergence. And then if populations are fully diverged, if they've been isolated for a long time, you can still see these signals of adaptive divergence, but they're likely kind of more hidden, kind of in the background of this high genetic drift. And so genetic drift is kind of just the random accumulation of genetic differences over time in isolated populations. And so I'm going to be talking a lot less about drift, but more about gene flow. So here these populations might still be kind of exchanging migrants. And so that's what we say when we mean gene flow. So gene flow is kind of successful migration in a genetics context. So clustering of adaptive variation in the genome prevents gene flow and recombination from disrupting gene complexes as they adapt. So this is kind of the why for the genomic islands of divergence. And I just, this is a really important concept, so I want to make sure to say it again. Basically, if these loci are in a small area, they're less likely to be disrupted by the forces of gene flow and recombination, which will break up these gene complexes. And the next couple of slides kind of illustrate this a little bit more. Okay, so again, gene flow can disrupt adaptive divergence. We think about gene flow often as a good thing in the context of like preserving genetic diversity and things like that. And it definitely can be, but in this context, gene flow can actually disrupt the process of selection and adaptive divergence because it introduces variants that may not be selectively advantageous for the population as it's kind of undergoing selection. Okay, so the important point here, though, is that if adaptive loci are far apart, the combination of gene flow and recombination can disrupt the sets of these loci. So in this little graph, let's say the yellowfish and the greenfish successfully mate. And in the yellowfish, there's these two red loci that are advantageous for adaptation. But when they mate, the resulting offspring have the correct genotype at this locus, but there's been recombination and they get the gray genotype at this locus. And so that's kind of undoing the kind of where selection is trying to go. But if we think about it this way, think about this time that these two red loci are very close to each other. And so again, we see the yellow and the green fish mate, but because these two loci are in tight linkage, that linkage block is maintained. And even though there's going to be signals of gene flow across a large amount of the genome from this green fish, these two loci have been preserved, and that genotype has been preserved, and those loci can kind of continue to evolve together and not be disrupted. So in other words, if loci are close together, recombination will be less likely to disrupt adaptive combinations, even when gene flow is high. And this is another important point that we're going to be coming back to multiple times in this talk. Structural variation like inversions can increase the size of these linkage blocks, making it possible to protect more adaptive alleles from recombination. So if you think about, and I'll explain what inversions are later, but let's say instead of this kind of area of linkage, an inversion makes it so like this whole piece of the chromosome is protected. Then those loci are kind of free to evolve without the kind of destructive or effects of recombination breaking up those complexes. Okay, so theory predicts that when gene flow is high, adaptive loci should be clustered in fewer genomic regions to protect them from recombination, but few studies have empirically tested this hypothesis. And so we had the opportunity to actually go and kind of test this hypothesis in the Mississippi River. This is a really neat project, a big collaboration between a bunch of agencies. These are a few of them: Wisconsin DNR, USGS, and then there's a great long-term monitoring program for the upper Mississippi River that we worked with on this project. And this project was led by our amazing postdoc, Yueshi, and the paper is now out as a from the cover paper in molecular ecology if you guys want to go take a look at it. Okay, so here's kind of where we're at here. We're in the Midwest. We're looking at kind of Missouri, Iowa, Illinois, Wisconsin, and Minnesota. And we sampled 6 different fish species from this area. So at each of these, sorry, I'll turn my pointer here. At each of these sites, we sampled, I think it was like 50 individuals from each of these different species. And these species run a kind of a large diversity, contain a large amount of life history diversity. And I'll get to that on the next slide here. Okay, so how different fish spawn has a lot to do with their kind of population connectivity, genetic differentiation, and gene flow. And one of the biggest kind of determinants of that is whether they're nest or broadcast spawners. And so, for example, bluegill are nest spawners. They don't have a huge home range. They don't migrate that far. They sort of stay about where they're at, and they spawn on nests, meaning that their larvae aren't free flowing and don't kind of get an opportunity to like move a long way down the river and kind of move a long way from where they were born. And you can contrast that with things like emerald shiner, gizzard shad, and freshwater drum, which are all broadcast spawners, meaning that their gametes kind of go out in the water column and they can be carried a long ways down the river. And that turns out to increase gene flow quite a bit as those larvae can kind of move a long distance. And then there's some kind of intermediates here. So both bullhead minnow and bluegill are nest spawners, and they're not really known to undergo large migrations. Channel catfish is a nest spawner, but they move around a lot as adults. And then emerald shiner, gizzard shad, and freshwater drum are all broadcast spawners, and freshwater drum is highly migratory as well. And so kind of our prediction here is that this life history is going to influence gene flow. So again, to test this hypothesis about whether gene flow kind of influences the genomic architecture of adaptation, we wanted to get species that kind of spanned a large range of gene flow. And this was kind of our prediction, which you'll see turns out to be pretty close to reality, is that these nest spawners are going to have low gene flow, meaning high genetic differentiation or FST values. The channel catfish might be kind of in the middle because it's a nest spawner, but it's migratory. And then you've got these pelagic broadcast spawners over on the high gene flow side. Okay, so just a quick kind of methods on the workflow. to assess gene flow and identify adaptive differentiation in the system. We genotype thousands of genetic markers in each species with rad sequencing, which is it's just a way to subset the genome down and get kind of look at a few thousand spots in the genome. And then we use multiple methods to identify putative, adaptive, and neutral loci, the outlier test type approach that we were talking about earlier. We assess neutral population structure and estimated gene flow. And then we placed adaptive loci across the genome. UA aligned all of the loci to different genomic resources, the closest sequence genome for each species, to place all the loci, or as many as possible, on the genome. And then we identified genomic islands and tried to look at the mechanisms that created them. Okay, so this slide should look pretty familiar. We had the prediction that I put up a couple slides ago, and this is the empirical results as far as FST. And so, like I said right in the beginning, kind of an FST anywhere near 0.1 is pretty high. And so our highest FST in the system was from bullhead minnow, which was 0.07, which it's pretty high, pretty close to that 0.1. Bluegill was also pretty highly structured with an FST of 0.03. And then we had a pretty major gap in between the next set of species, which all had FSTs below 0.01, which is pretty low. And these three, drum, channel catfish, and gizzard shad, you would probably call that structure, maybe detectable, kind of right on the range of like something that would be statistically significant. And then emerald shiner, the structure was extremely, extremely small at neutral loci. Basically, we can call this, panmictic at neutral loci, and panmictic means essentially no genetic differentiation. Okay. So this is kind of the meat of this, so I want to go through the slide pretty slowly. And this is all the genome scans, meaning the FST across the genome for each of the different species. Again, FST, or genetic differentiation, is on the y-axis, and each different chromosome, or the genomic position, is on the x-axis. Each different dot is a marker. The dots with orange are statistically significant outliers. And then these blue areas are trying to recognize different windows or pieces of the genome that are statistically kind of differentiated and probably putatively under selection. Okay, so first with the bullhead minnow, remember we said this was the highest genetic differentiation in the study. So it was high differentiation and few notable adaptive loci or islands of divergence. Next we have bluegill. So intermediate differentiation is still pretty high. And what we see here, we see a lot of small islands. So frequent islands with kind of a small number of loci in each island. Okay, next we get into freshwater drum. So we're seeing, as we move kind of up in gene flow, down in FST, we're seeing less islands, but the islands are getting kind of more prolific compared to the background genomic kind of differentiation. So we only see like 3 or 4 islands here, but they're actually getting to be pretty major outliers in the context of the rest of the genome. Like this one here, or this one. Okay, moving to channel catfish. Again, we see few but prolific islands, something like this over here. That's a pretty high outlier compared to the rest of the genome. Same with this and this. Gizzard shad really didn't show a lot. It just didn't have a lot of islands, not a lot of evidence of adaptive differentiation here. And then moving to kind of the lowest differentiation, highest gene flow, and probably most interesting result here is that, remember, this emerald shiner at neutral loci, meaning loci that are not adaptive, that we don't think are involved in adaptive divergence, it showed a infinitesimally small FST of 0.0003, meaning that if we just looked at those neutral loci, we'd say there's no way that there's genetic differentiation in this species. But on chromosome 9, you can see this huge piece of highly differentiated loci. It's actually 42 megabases, which is, was that 4.2 million bases, and it is 40, yeah. And it actually turns out to be an adaptive inversion. So we'll get into what an inversion is in a little bit. But this was kind of a really neat result where just as we predict, when genetic differentiation is extremely low, there needs to be kind of a structural mechanism to isolate loci under selection to prevent them from kind of being eroded by recombination. And indeed, we see in our lowest FST species a large island of divergence corresponding to a inversion that's likely kind of isolating really important loci, so this fish can evolve even under extremely high gene flow. Okay, so just talked about a lot about inversions. Here's what they actually are. So inversions are a common structural variant where segments of DNA flip 180 degrees and are reinserted into the chromosome. And this is the most important piece for kind of why inversions are important for kind of adaptation is recombination across inversion types is rare, as recombinant gametes are generally inviable. And so what that means is that genes found on inversions are shielded from recombination, facilitating adaptive divergence even when gene flow is extremely high. And actually the cod example I showed you earlier, that's another example of inversions. And so as right now, theory is essentially predicting that when gene flow is extremely high, really one of the only ways that you can get kind of this adaptive divergence and these islands of divergence is through structural mechanisms like these inversions. And so again, an inversion is an area of the genome that's flipped. It means that recombinant gametes are inviable. You can look at the diagram, it's kind of complicated, but basically those gametes are inviable. And that means that genes found on inversions are allowed to kind of adapt without being broken up by this recombination. And so these inversions look to be a very important way that populations with extremely high gene flow can adapt to kind of variable environmental conditions. Okay, so how do we actually get evidence that this thing is an inversion? And so there's a couple ways that we can look at. And so first, when you do a PCA of this region, usually what you want to see is kind of 3 discrete genotype clusters. And so we've got a homozygote over here, heterozygotes over here, and then the alternate homozygote here. You like to see more heterozygosity in the heterozygote. That's pretty obvious. And so we see higher heterozygosity. Another really good way to look and see if something's an inversion is that it's going to have more linkage disequilibrium across genotypes than within one of the homozygotes, and that's what we see. And so linkage disequilibrium is essentially the non-random association of genotypes at various loci. So basically, like you can think about like the correlation between genotypes. So if it's an A over here, it's an A over there. If it's a B over here, it's a B over there, and we can always kind of that relationship is what linkage disequilibrium is kind of describing. And so we see this where we see high linkage disequilibrium across the genotypes, but low within the homozygotes. And these are the actual frequencies of the different genotype classes at the inversion. And so you can see here, it goes from north to south. And you can see that in the north, it's fixed for this blue homozygote here. And then as you move south, you start to get a lot of heterozygotes and then some of these alternate homozygotes. And so there's a big frequency difference in these genotypes, even when the rest of the genome is showing really no genetic differentiation. Okay, so conclusions on the Mississippi River fish. Life history diversity, or life history influences genetic structure. Species with lower gene flow contain more and smaller islands of divergence than fish with higher gene flow, with the most extreme example being those emerald shiner, where there's extremely high gene flow, FST of basically 0 at neutral markers, but this really prolific adaptive inversion that's probably facilitating adaptive divergence in this fish. So yeah, at the extreme, emerald shiner were only differentiated at a large inversion and not really anywhere else in the genome. And so our study provided rare empirical evidence to support the theory that gene flow influences the genomic landscape of adaptive divergence. That is, one more time, that in In low gene flow species, you're going to see more but smaller and less prolific islands, whereas in high gene flow species, you're going to see less and more prolific islands of divergence. Okay, so the next steps are kind of, or at least where we're going in this talk, is trying to understand a little bit better the mechanisms that create islands and what conditions favor certain mechanisms. And so I kind of said before that we're going to get back to these sockeye, and that's what we're going to do here. We're going to use sockeye salmon ecotypes as a model to understand islands of divergence. Okay, so like I said before, I spent my PhD studying sockeye salmon in Southwest Alaska. Here's just a picture of me, I think, from my first or second summer, and then my fifth, really embracing the Alaskan lifestyle, which ended up with me actually moving back. And so sockeye salmon have different biological features depending on the habitat they occupy, leading to the formation of distinct ecotypes. And kind of the most conspicuous example of this is sockeye salmon that spawn on lake beaches versus that spawn in small streams. And so sockeye that spawn in lake beaches are not really under selection from, or under pressure from predation and kind of just depth of the stream. And so they evolve these really large, kind of deep-bodied phenotypes due to sexual selection, whereas sockeye that spawn in these small streams are much more fusiform, much more like skinny or streamline shaped because of the kind of just depth limitations and predation by bears. And so you'll see if you like, I should have taken a picture. Maybe I will when I go back up. But if you try and if you see a beach ecotype trying to stray into one of these streams, it's just like it flops over, and then the seagulls like pick its eyes out immediately. And so it's pretty much like watching, you know, selection in action. There's it's very hard to migrate. If you're a beach fish, it's very hard to kind of migrate into these small streams. So here's kind of more pictures, a little bit clearer pictures of these different ecotypes. We're going to look at beach, stream, and then river, which is a little bit different. It looks more like a beach, but there's some kind of major environmental differences there too. And again, these are the habitats that they spawn in. Okay, so a couple papers from my PhD identified islands of divergence in these sockeye salmon ecotypes, and I'll show a little bit more about that. So first, we went to the Wood River system, which that's where those pictures were from. It's in Southwest Alaska. And we looked at 6 populations, 2 from each ecotype. And then we did rad sequencing and put a few thousand loci on a genetic linkage map, which is just a way to place those loci on the genome. And here's the results from that. And essentially what you can see is that most of the genome is showing very little genetic differentiation, whereas there's a couple places that are showing extremely high differentiation. Again, these islands of divergence. And so if we kind of go back to the last kind of species we talked about, sockeye salmon in the system have a neutral FST of about 0.01. So they would have been like in between the bluegill and like the next set of species down. So definitely quite a bit of gene flow, but not as much as like the emerald shiner, for example. And so we see that most of the genome shows pretty low genetic differentiation, but there's a couple like really prolific islands, most notably here on chromosome 12 and chromosome 13. So again, that study was just these 6 populations in the Wood River, and we wanted to expand that to try and see if those islands showed up again in different drainages. And so we looked at Iliamna, Chignik, which drains down into the North Pacific, and then the Copper River. And we don't need to worry about the purples too much here. And so we looked at less loci here, but we looked at those different drainages, and we see Wood River, same deal. The same islands pop up. Iliamna, they're repeated. Chignik, same islands, and then the same in the Copper River. There's more kind of background genetic drift, genetic differentiation here, but the same islands still pop out. So it looks like these islands of divergence that we found in sockeye are conserved across study systems. Okay, so where we're at right now is that we've done some kind of low density sequencing, rad sequencing, and even fewer loci. And we found that the SNPs in islands of divergence, or the genetic markers in islands of divergence, show high differentiation across drainages. But what we don't know is the full landscape of adaptive divergence, the genetic mechanisms underlying these islands, and the evolutionary history of islands. And so where we went here is we went to genome resequencing. And so everything I've shown you so far has been rad sequencing, and that's been 5,000, 10,000, 3,000, thousands of markers up to maybe 30,000. And genome resequencing allows you to genotype nearly all the markers in the genome rather than just a subset or a few percent. And what that does is that really cleans up kind of the picture of what's going on when you're trying to kind of understand these islands of divergence. And so kind of a good analogy is like you get this sort of pixelated picture where you can, there's a flower there for sure, but like, you can't see the petals and the shape and all that. And then genome resequencing is more like this really clear picture of the flower. And so we moved from, about 3,500 SNPs here to 2 million. And we looked at 7 different populations, 3 populations from the Wood River that we've seen before, 3 populations from Iliamna. I'm going to focus on these 2 systems. And this is sort of an outgroup of river type sockeye salmon that I'm not going to get into too much. And we did about 30 populations, 2 million SNPs. And for the kind of folks really tuned into whole genome, sorry about that. We got about 7 or 8x coverage to call genotypes. Okay, so here is the kind of raw results of this search for islands of divergence. And what we can see here is that there's some conserved islands and there's some kind of islands that are only found in a given drainage. So for example, this island on chromosome 22 is really only found in the wood. It's not very prolific in Iliamna. Kind of the same thing for this 14. It's only found in Iliamna, but not really found in the wood. And so we wanted to try and focus on here are islands that are conserved between the 2 drainages. And that turned out to be the island on chromosome 3, chromosome 5, 12, and then chromosome 13. And just kind of to point out, some of these FSTs are pretty bonkers. You're talking about FSTs above 0.6 when the neutral FST for both of these systems is about 0.01. So some really prolific islands kind of on the background of a pretty low FST. Okay, so we identified these 4 islands that we wanted to check out, and we wanted to really look into which mechanisms created these islands. Okay, so again, we talked about inversions, and I'm not going to reread this slide, but basically we're thinking about, are these islands inversions or are they caused by something else? And so we talked about inversions, but another mechanism that can cause these islands is called divergence hitchhiking. And that's gene exchange between diverging populations, when gene exchange between diverging populations is reduced around a gene or genes under strong divergent selection. And the underlying recombination rate is not impacted, but between population recombination rate is reduced due to selection. And so in other words, this mechanism does not require structural variation like the inversions. This mechanism just kind of relies on strong selection to select against kind of recombinant individuals to the point where kind of between population recombination or between population exchange of genes is reduced due to selection. And so the real goal here is kind of trying to differentiate between whether an island is an inversion or whether it's this divergence hitchhiking. That's at least our 2 kind of main hypotheses. There's also some other ways that islands can evolve. Okay, so how to use genomic data to investigate island mechanisms. And we talked about linkage disequilibrium briefly before, but again, for linkage disequilibrium, inversions should have relatively conserved linkage blocks with discrete endpoints. PCA inversions should have 3 dominant genotype clouds corresponding to genotypes, and those should be conserved across a large geographic area. And then genotype heat maps, inversions should have a clear conserved genotype blocks. And so this kind of is related to what I showed for the Mississippi River one, where we looked at linkage disequilibrium and we looked at principal component analysis. We didn't really have any of these heat maps in there. Okay, so first off, let's look at linkage disequilibrium in the 2 drainages. So this side is Iliamna, this side is the wood, and each different plot is a different island. And so some things are probably popping out pretty quick that like most of the islands have pretty conserved, pretty large blocks of linkage. And probably the best example here is 12. So 12 has like a large, consistent block of linkage with kind of discrete endpoints. And just again, the red here is higher linkage, and then the blue is lower linkage. So this is kind of a linkage block where all the genotypes in this region are very similar. Very similar for a given, associated with each other. And we can contrast that with chromosome 13, which has very patchy linkage. So like you'll see some red stuff over here, but then there's some blue, but then there's some red. But then there's a very small conserved area kind of right over here, and it's right over here again. Okay, and next we're gonna zoom out. I mentioned a little bit kind of that things should be conserved across a large range. And luckily, when the Coop Lab and Chris Christensen did the sockeye salmon genome, they did genome resequencing on about 100 individuals from across the range of sockeye salmon, from the Columbia and Fraser all the way up through Bristol Bay and then Russia. And so we could merge those data with our data to try and understand kind of the frequency and the kind of characteristics of these genomic islands of divergence across the sockeye salmon range. And so here are range-wide PCAs. And again, none of these are quite as clean as the Mississippi River one, but a couple of them get pretty close. And so first off, chromosome 3, it's interesting that if you just look within the Wood River, which is like kind of these dots over here, you start to see 3 clouds, but then it gets really messy when you include populations from the Fraser and further south. Okay, chromosome 5, looking really good here. We've got 3 discrete clouds with some subvariation within inversion types, which is common and kind of, we didn't see it too much in the Emerald China, but it's pretty common, especially with these smaller inversions. And then chromosome 12, we see again 3 nice discrete clouds, and then 13 is just like a junk show, super messy, like you can't really even tell much at all. So very, very messy. And sorry about that. And I think the big point here is contrasting something that looks like this or like this, but especially this, with something that looks like this. So it's pretty clear that like there's kind of a different something going on here versus there. Okay, so here's genotype heat maps, and basically the different colors correspond to different genotypes, with orange being heterozygotes, this kind of yellow being one homozygote, and the red being the other homozygote. One thing that's interesting to note here is that the individual where the genome sequence was produced from is in this brown cloud. And so for chromosome 5, it's a clean homozygote here. Same for chromosome 12. And you can see here that 12 and 5 are conserved really well across the whole range. You see, like, for the most part, all the heterozygotes are going here, all this alternate homozygote is here, and then all this homozygote is here. And you see consistency in the reference individual where, you know, for the reference populations, it's a clean, consistent reference allele with no swapping. Chromosome 3, we see, like, so again, the reference individual is this brown, and we see kind of swapping here where, like, sometimes it's this allele, sometimes it's that allele, and in general, it just looks a lot less clean. And then kind of the same thing with chromosome 13, where we don't get these clean blocks in the way that we get it with 5 and 12. Okay, so I think you've probably seen that I'm heading this way, but it looks like the islands on chromosome 5 and 12 are likely caused by inversions, whereas the ones on 3 and especially 13 are probably more likely to be divergence hitchhiking. One thing I didn't talk about too much here is that in 13, you have to zoom in very at a very fine scale, like 30,000 base pairs, to get to this small region that looks conserved across the range, whereas on 5 and 12, the conserved regions are somewhere in the like 80,000, 100,000, 150,000 base pair range. So still a lot smaller than that kind of multiple millions for the Emerald China, but a lot higher, a lot bigger area than for 13. And so sockeye inversions seem to be small compared to many other systems like cod or Emerald China. And our study shows that these small inversions can also be evolutionarily important. and that there's low but non-zero genetic differentiation in our system that likely creates suitable conditions for both types of islands within the same genome. So this is kind of interesting. Like for Emerald Shiner, we only found evidence for that one large inversion, and there was very, very high gene flow, very low genetic differentiation. And it looked like in that genome, there was really only evidence for that inversion. But the fact that sockeye have a little bit higher but still low genetic differentiation, it looks like that leads to both islands, or leads to both inversions and divergence hitchhiking, creating islands within the same genome. And just a caveat, while our mechanistic evidence is compelling, a long-read sequencing data would be necessary to sort of confirm 100%, and we didn't do any of that here. Okay, so with the last little bit of my talk, I want to talk more about kind of the so what of all this research. So first off, islands of divergence represent evolutionary shortcuts that take advantage of standing variation to achieve a desired phenotype rapidly. And so conserving diversity at these islands will allow populations to respond to novel stressors. And understanding the mechanisms that create islands will allow for more effective management of diversity. For example, developing a broodstock management strategy that targets and preserves important adaptive variation. And so I just want to step back a little bit and think about kind of different systems and where adaptive genetic data are really useful for kind of managing those systems. So I made this little tables with sort of different types of differentiation. So like, is the species, are they differentiated by habitat, by phenotype, adaptive genetic diversity, and neutral genetic diversity? So for sockeye, you can see just based on like ecological data that they have different habitats, different phenotypes. And then we've added this adaptive genetic diversity component, whereas at neutral markers, there's really very little to no genetic differentiation. And so the phenotypic diversity in the system was apparent before genetics, but genetics provided additional evidence that this diversity isn't plastic and maybe may not re-evolve quickly. So another system here is the Emerald Shiner that we looked at. And so we would assume that they have different habitats. So remember, the northern populations were fixed for a single genotype at that inversion, whereas the southern populations had mixtures of both genotypes and heterozygotes. And so we think that there's adaptive divergence between kind of the northern and the southern populations. And so presumably these fish live in different habitats, north versus south. We have no idea if there's phenotypic differentiation. There's no neutral differentiation, and there definitely is adaptive genetic differentiation. So basically, important adaptive diversity was completely cryptic before the genetic results. Our genetic results show that for some reason in this fish, there's this large inversion that is highly differentiated between the areas, and so there must be, or there's very likely to be adaptive differences between these areas. Okay, so the next one here is that these different phenotypes of cod that our postdoc Sarah actually has studied quite a bit, and so there are these different colors. We don't know if they spawn in different habitats. We know there's phenotypic differentiation associated with these colors, and it actually turns out that there's no adaptive genetic diversity and really no neutral differentiation either. And so sometimes phenotypic data does not correlate to adaptive genetic diversity. And genetic data here provides confidence that phenotypes can probably be managed together. You don't have to try and have like a different assessment for the olive versus red cod, for example, at least it seems like. And now let's contrast that with kind of the classic example of cod, which are these offshore and coastal ecotypes. And so for these Atlantic cod, there's populations that kind of live offshore, migrate a long ways, and there's populations that are more coastal, associated with fjords. And they have very different kind of ecology and life history and population dynamics. And so we know the habitats are different. There's not really any phenotypic or any clear phenotypic differences based on just looking at it. But there's very, very high adaptive genetic differentiation on the background of no structure. And so here, adaptive genetic data illuminated genetic differences between ecotypes. Further investigation revealed different ecology and population dynamics. And these data are now used to manage the stocks discretely in some areas. So this is a situation where kind of the adaptive genetic diversity and understanding that differentiation was extremely key to effectively managing these populations of cod. Okay, so just kind of zooming out a little bit here. Sometimes neutral and adaptive genetic diversity corresponds to conspicuous life history variation, talking about sockeye salmon, Chinook salmon. Sometimes it's cryptic, in the example of Atlantic cod. Sometimes phenotypic diversity is not correlated to adaptive genetic diversity. And so there's kind of all these different situations. And one other thing that's really important to realize is that observing the environmental factors that could create adaptive diversity is extremely difficult in some environments like the ocean. And you can think about the example in Emerald Shiner, like, yeah, there's definitely differences in the environments between North and South, but we didn't know if that was going to create adaptive diversity or not until we looked. And so genomics can help shed light on this important diversity without requiring direct ecological observations. And then hypotheses based on genomic data can then be tested. So for example, with the Atlantic cod ecotypes, you'd see this big adaptive genetic diversity, and then you could go kind of track that down with like tagging and kind of like fatty acids or isotopes or a bunch of different ways to see how different those phenotypes actually are. So how do we promote resiliency in the face of a rapidly changing environment? And this is kind of shifting back to what we think about a lot here in Alaska at the Alaska Fisheries Science Center. A lot of headlines like this, coastal Alaska cod are disappearing. There's been big warm blogs, big marine heat waves. Crab stocks are in kind of a decline in a warming Bering Sea. Marine heat waves are supposed to kind of impact things in a pretty major way. And so how do we promote resiliency in the face of this rapidly changing environment? And so to think about that, I just wanted to bring back an example from Bristol Bay. And so again, a robust portfolio of adaptive diversity is vital for resiliency. And this is a really interesting example. So these different colors are the different stocks of sockeye salmon in Bristol Bay and their abundances over about a 100-year time series. And you can see the yellow is this eggigic stock, and it was a pretty small producer for most of this graph. But then there was this big regime shift, and for some reason the eggigic stock just started to take off due to probably changes in marine conditions. And so the really important point here is that a stock that may be weak today could be strong tomorrow. And so kind of understanding our portfolio of adaptive diversity and making sure that we preserve all of these important adaptively different stocks is vital for making sure that we have the stock that's going to be successful tomorrow, and we don't take it out before it's kind of ready to shine, I guess would be a way to say it. So our goal at the Alaska Fisheries Science Center is to identify and help preserve important adaptive diversity to promote resiliency in some of the world's largest fisheries. And there's been some work already in our group. This is a great paper that Ingrid published a couple months ago. And there's a lot more work happening. And you guys are going to hear about that a lot over the next few months here. So I think we have the next 3 months of these omics seminars. Diana's going to talk about eDNA next month, and then Laura is going to talk about rockfish, and Ingrid's going to talk about gattids, so cod and pollock. And so I'm really excited to hear what they have to say, and hope you guys tune in so you can kind of see how we're integrating genomics and omics into management of Alaskan fish species. So looks like I took up almost exactly the whole hour here, but thanks so much to all the collaborators and funding sources for all this work. And oh yeah, these great sockeye photos are from Johnny Armstrong and Jason Ching. They have amazing photos online if you want to check those out. And then with that, I will take some questions. Thanks so much, everyone, for listening today.
Awesome. Thank you so much, Wes. What a fantastic presentation you gave today. And thank you so much for plugging the next few months of talks. We do have tons of folks from the Alaska Fisheries Science Center who will be giving talks in our seminar series. So everyone be on the lookout for that on our Omics website. So we do have like a minute or so to answer some questions. We had 2 questions that came in. So our first is from Sarah. What genes are associated with the putative inversion in shiner or sockeye?
Yeah, so this is where it gets hard because 42 megabases, there's a lot of genes in there, and it's hard to know which ones are actually the important ones, in other words. So that's one really tricky thing about inversions is that, like, you know, if an inversion is half a chromosome long, it's hard to pinpoint the gene. So we looked in Emerald Shiner. We didn't see anything that was like, you know, screaming out as something super functional. I will say there's a gene on the chromosome 12 in sockeye called LRCC9 or something. I don't remember what it does. It usually they come back and say they're like a cell signaling pathway or something like that. I will say chromosome 13. Oh, sorry about that. Chromosome 13. It looks like that really conserved region is right around a gene that's involved in, it's like an estrogen receptor. And we don't know why. So I kind of purposefully avoid going too far into the gene annotation thing because it's just kind of hard to really know for sure what genes are involved. The next step would be kind of, you know, looking at those genes and trying to see if you could kind of manipulate genotypes of those genes to see if it has an impact on phenotype. And some of the salmon work with like VGLL3 and GREB1 has, well, mostly VGLL3 has started to do that, but it's pretty tough. So sorry, not really a super fulfilling answer there, I suspect, but that's kind of the journey.
Great, thank you so much, Wes. We are a little over time. I do have a few other questions that have come in. So I think what I'm going to do is the folks who did ask questions, Wes has his email up here on the slide. So I encourage you to please just reach directly out to him. And if you are on my NOAA Omics email list, his email should be on there now. But I do want to take a moment again to thank you, Wes, so much for presenting today and to thank everybody for joining the seminar. Just a couple quick reminders that our seminar takes place on the third Wednesday of every month at 12 p.m. Eastern time. And our next seminar is going to be on March 16th. Our presenter is Dr. Diana Batcher, and she's presenting on a talk titled Measuring the Attenuation of Marine eDNA in Nearshore Alaska. So the registration link is currently up on our website. So thank you everybody again for joining and have a fantastic rest of your day.
Thanks everyone. Thanks, Catherine.